Hi guys, we're going to be doing a presentation on gene editing and neurodegenerative diseases and how genetic engineering has been helping with solving this problem. We're going to start off with the history of genetic engineering so you can have a background for what's going on currently. It starts off in the 1950s, the discovery that DNA is a double helix. This was extremely important because it allowed, since researchers now knew the structure of DNA, they were able to research on it more and figure out how to replicate it. 1958, DNA is made in a test tube for the first time. 1962, there is a special jellyfish protein that is isolated and can be used with another gene that can determine which cell is expressing the gene that you want to see what characteristics it has. This was extremely important because people can now identify which genes are causing which processes. In 1967, DNA ligation links DNA fragments together. This is obviously very important because this paved the way for many experiments in the late 70s, particularly recombinant DNA. In 1968, there was discovery restriction enzymes, which helped cut DNA, which allowed multiple DNA things to be spliced together at a later point. Next slide. In 1971, gene splicing and paving the way for recombinant DNA because of this researcher named Paul Berg. He became the first scientist to create recombinant DNA from more than one species. He cut the DNA from two viruses, creating sticky ends. Then the ends would anneal on their own after incubation, and the addition of the DNA ligase would seal it. And this proved that it was possible for any two DNA molecules to be joined together, which is a huge stepping stone for modern recombinant DNA. In 1971, type 2 restriction enzymes were used to map some DNA sequences. In 1972, recombinant DNA was first created. It's DNA that's been created due to the combination of elements of DNA from different organisms. And this truly happened the first time and was probably one of the biggest breaking points. The next slide. In 1981, the first transgenic animal transferred the gene of a rabbit into a mouse. In 1982, the first synthetic insulin drug was created. This was extremely important because insulin was extremely expensive, required a lot of materials and animals, and it was very inefficient. But with synthetic insulin, this suddenly became a lot easier and it was more widespread and more people could have access to it. In 1983, PCR was developed. This technique was extremely important because it promoted the idea of backwards genetics. With forward genetics, scientists generate random mutations looking for something significant, but it's possible to not find anything and it's more of a guess and check method. But with backward genetics, they're able to identify the exact gene causing the mutations. In the 1990s, the first clone of an animal was made, cloning a dolly of a sheep. It had the exact same genetic identity, which was a huge feat. In 1999, the first human chromosome is sequenced, though it fully wasn't complete in 2003. Scientists demonstrated they completely mapped chromosome 22. The full chromosome sequence is extremely important, especially for human genetics and trying to cure diseases in the human genome. Next slide. In 2000s and onward, in 2001, the first gene-targeted drug therapy is approved. This is a huge stepping stone because this is the first actual genetically engineered type of thing that is really allowed to be with the public, and it's a huge stepping stone. In 2006, FDA approves the first preventative cancer vaccine, which also happens to be gene-targeted. In 2010, the first synthetic life form created by Craig Venter. This is an important milestone because it proved that life can be created synthetically, which has a great number of applications in genetics. 2012, CRISPR genome editing tool is created and visualized by Jennifer Luna and Emmanuel Charpentier and their teams. This is now the most common form of genetic engineering, and it is probably the most important thing in genetic engineering at the moment. 2015, the first GMO salmon is sold to Canadian markets. This is important because it provides a solution for the world's overfishing crisis and provides an example of how genetic engineering can solve many different problems around the world. In 2015, human embryos at new CRISPR. Chinese researcher known as Yunji Huang did this, and it became an ethical controversy. This was 3 years before human trials for CRISPR were approved, and in 2018, they were finally approved.
So next is gene editing methods. In general, gene editing methods consist of a guiding agent and a cleaving agent. The guiding agent identifies the target site on the genome. Then the cleaving agent breaks the DNA strand at the site the guiding agent identified. The three most common gene editing methods today are ZFNs, TALENs, and CRISPR. The ZFN, or zinc finger nuclease, is a manufactured complex of a zinc finger DNA binding protein with a restriction endonuclease called foci. The zinc finger protein identifies a cleavage site on a DNA strand that is 3 to 4 base pairs long. Usually longer user-defined sequences are needed to be identified, so a chain of zinc finger proteins is specially manufactured. The chain is attached to the endonuclease, which cleaves the DNA at a specific codon in the target sequence. Next, TALENs are very similar to ZFNs, with the major difference being that the tail protein, which binds to the DNA, can only recognize one nucleotide as opposed to zinc finger proteins being able to identify 3. Again, like ZFNs, chains of these tails or transcriptor activator-like effector proteins are specially manufactured to identify a user-defined sequence. Then, the foci endonuclease, like with ZFNs, cuts the DNA at the specified spot. Finally, CRISPR, the bacterial antiviral defense mechanism, stands for clustered, regularly interspaced palindromic repeats. These repeats are copies of sections of viral genomes stored in bacterial DNA. Bacteria use these repeats to identify invading viruses. Then they give the Cas9 endonuclease a guide RNA or gRNA with a transcription of the segment of viral DNA, and the Cas9 enzyme will chop up the virus at those sequences in the genome and destroy the virus. However, a limitation of the CRISPR-Cas9 mechanism is that it only works on sequences where there is a protease spacer adjacent motif, or PAM, next to the target sequence. Different endonucleases have different PAMs, but Cas9's PAM is typically any base followed by 2 G bases. The gRNA guides the Cas9 to the PAM on the DNA. The Cas9 enzyme then cleaves the DNA 3 base pairs upstream of the PAM. This method of gene editing is also sometimes called RGen, standing for RNA-guided endonuclease. In this table, ZFNs, TALENs, and RGens are compared by manufacturing process, cost, specificity, off-target activity, efficiency, possibility of immune reaction, and clinical and research applications. RGens are the easiest to manufacture because the only portion that is unique to each different use is the guide RNA. For ZFNs and TALENs, multiple guiding agents need to be strung together in different patterns for each different use. However, some say this makes them less likely to cause off-target mutations because each piece has a higher chance of doing its job properly compared to the entire 20 nucleotide long guide RNA for an RGen. Except for the possibility of immune reaction, RGen beats ZFNs and TALENs in every other category listed, making it easy to see why, despite imperfections, they're the most popular gene editing method right now. Some other less common or obsolete gene editing methods include conventional homologous recombination, peptide nucleic acid systems, adeno-associated virus systems, and meganucleases. Computational biology and gene editing. When picking a target sequence in the genome to edit, there are many factors to consider: location, length, suitability for the guiding agent, potential for off-target events, and more. There are online tools that will find a target sequence based on given parameters. There are also tools that will predict places in the genome where off-target activity is likely to occur. We'll look into 2 commonly used tools, ChopChop and Cas-Finder. First, ChopChop, made by Harvard University, is an online tool that finds target sequences. The input is a target gene in the form of the gene name, its genomic coordinates, or the DNA sequence. The organism whose genome will be edited, and the editing method, including the nuclease type for CRISPR, and the purpose of gene editing, for example, to knock out a gene. The tool then uses bow-tie alignment, which is a fast, memory-efficient alignment program for short DNA reads to find suitable target sequences for the given input. Next, Cas-Pathfinder, made by Hanging University, Korea, finds possible regions in the genome where off-target mutations could occur. It takes in a PAM type, the target organism or genome, and the query sequence, which is the target sequence meant to be edited. It returns a file of information like position and number of mismatched bases on each sequence it found where off-target mutations could happen. Using the SP-Cas9 editing method as an example, Cas-Pathfinder finds all 23 base pair long sequences in the genome that are similar to the 20 base pair long guide RNA in the 3 base pair long PAM. Unlike other similar tools, Cas-Pathfinder allows for variations in the PAM sequence. Then it finds the number of mismatches of all compiled sequences when compared to the query sequence. Only the sequences whose mismatch count is under a threshold will be outputted. This process is done on chunks of the genome simultaneously.
So our next topic is applications and examples of gene editing. Next. First, what is gene therapy? Gene therapy is a technique of modifying genes into patient cells to treat diseases or disorders. The goal of this is to directly correct the mutations that have occurred within the DNA instead of using or resorting to drugs or pharmaceutical methods. Through gene therapy, numerous effective treatments have been proposed, some of which achieved against many genetic disorders. Next. In a gene therapy trial at UCSF, Dr. Christoph Bankiewicz worked with 7 children from ages 4 to 9 who were born with a deficiency of AADC, an enzyme involved in the synthesis of neurotransmitters regarding movement, mood, learning, and concentration. These children were unable to speak, express, or feed themselves, or hold up their heads. Next. Bankiewicz developed a viral vector containing the AADC gene, which was infused into the brain shown in the picture here. With real-time MR imaging, his team was able to find the target region, the brainstem. Next. After completing this part, over a few months, the children's conditions improved drastically. As quoted by the article, they had improved motor function, better mood, and longer sleep, and were able to interact more fully with their parents and siblings. Many began laughing, speaking, and even walking. Before the trial, many underwent oculogyric crisis and involuntary fixed gaze, with seizure-like episodes that could last for hours. This crisis went away in all but one patient. This trial is one of the many examples of gene therapy that have brought success to treating disorders. Next. So here's a video that we'll watch a few seconds of. So this is a before and after comparison video of the successful results of gene therapy in this trial at UCSF. You can directly see the great impact that gene editing has had on humans here. And yes. Okay, moving on from viral vectors, I'll be explaining the applications of a few methods that Siri previously talked about. First, ZFNs. Studies in Taiwan have effectively applied this method to Parkinson's disease. A missense mutation is a change of a single base pair that results in a different amino acid in the protein. With this mutation in the critical SNCA gene, which would normally release neurotransmitters that relay signals between neurons, symptoms arise in patients. These include tremor and rigidity that could spread and progress to dementia and hallucinations due to the loss of neurons in the midbrain. Current treatments have temporarily slowed these symptoms, but no complete cures have been found. As the SNCA gene is critical for normal brain function, this team used induced pluripotent stem cells, or iPSCs, to find a gene editing treatment. iPSCs are excellent for gene therapy purposes as they're derived from skin or blood cells to enable the development of any type of human cells. Using these cells and the ZFN method, they found out that the length of neurites in corrected iPSC cells were longer than those without corrections, which significantly helps alleviate the effects of the SNCA mutation, allowing for more opportunities and research to be done regarding the cure of the Parkinson's disease thanks to this method. Next. Okay, next, talons. This method was used with Huntington's disease, which includes motor dysfunctions, behavioral or psychiatric disturbances such as cognitive decline, personality changes, poor attention, and dementia due to a mutation in exon 1 of the HTT gene. Like Parkinson's disease, there is no effective treatment yet. However, the same team in Taiwan worked on using talons towards HD. Through gene editing, this treatment reversed the mutation's effects. Corrected iPSCs had lower susceptibility to cell death and increased productivity with cellular energy production in neural stem cells. This method has been proven to be powerful but also slow with the large size of DNA and its inefficiency with targeting multiple DNA sites simultaneously. Next. Last but not least, CRISPR, one of the most popular gene editing tools nowadays. Dr. Nicholas Marianne's team used this method also on HD in mouse models and human iPSCs. gRNA is guide RNA that recognizes the target DNA region of interest and directs the Cas nuclease there for the actual gene editing. They use this to recognize the mutant HTT gene as well as the Cas9 protein to start the DNA repair process. During their study, they found that if they permanently halted the mutant HTT gene in mouse cortical neurons or human iPSC-derived neurons, symptoms of HD significantly alleviated. Next. Looking into their data, they realized that mHTT, or mutant Huntington protein expression leads to the loss of HTT, resulting in embryonic lethality. They did this with CRISPR, lowering embryonic lethality and alleviating motor deficits caused by the disorder. Let's look at this graph. The black line, WT, represents wild-type mice, which are the standard normal individuals. The blue line Ki-controlled gRNA refers to knock-in mice, which have altered gene sequences to help scientists study specific genes. The red line Ki-HTT gRNA refers to the same thing, but this gRNA has been corrected with CRISPR. A rotor test evaluates motor coordination, balance, and motor learning in the mice. The wild-type mice showed the longest duration of motor control during this test, which makes sense as they did not have the mutant HTT gene. But over the span of 12 weeks, we can see that the corrected gRNA, the red line, indicates a higher rate of success than the uncorrected gRNA, the blue line, as these mice were able to successfully demonstrate motor control for a longer duration. This improved performance alleviated the motor deficits of HD in those mice. Hence, CRISPR is very advantageous in correcting gene mutations. Not only does it open up opportunities for treating neurodegenerative diseases by inhibiting or modifying mutant protein expression, but it also is also a cheaper method that can simultaneously modify genes at multiple independent DNA sites. Next.
Okay, so the final topic of gene editing is the ethics of gene editing. Next slide. Okay, so there has been controversy surrounding gene editing for many years. Most of these stem from basically when it was being first introduced, maybe like in the early 21st century. So there are religious concerns. Critics believe that scientists are essentially playing God. There are also other concerns such as designer babies. Critics also believe that we're on the path of designer babies, which are basically babies that have certain characteristics, cosmetic characteristics that are desired by the general public. And as you can see in the graph on the right here, 94% of people, this is a survey from 2004, believe that that is going too far. Critics believe that gene editing is unsafe. This is more of an older opinion, like 22 years ago. Jesse Gelsinger died from a rare liver disease while gene therapy was being experimented on. There was a gene therapy experiment at the University of Pennsylvania, and now it is a lot safer since the introduction of CRISPR. It's become a lot safer. Next. Okay, so there are two types of gene editing. Somatic gene editing, which is the ones my group mates were talking about, which is basically targeting a specific cell in an already formed organism, and it won't have any effects on future generations. Well, germline gene editing has effects on future generations, and it targets the cells early in development, or like an early embryo, for example, for a human. And that brings me to my next point. Next slide. There was a controversy with the gene editing, with germline gene editing. So in 2018, at the Second International Summit of Genome Editing, there's a scientist who have claimed to gene edit on 2 embryos, which is germline gene editing, because they are experimenting on the organism early in development. And there is a lot of outcry from many scientists across the world. Robert Troge, director of the Center for Bioethics at Harvard Medical School, says, "To me, the controversy around Dr. He is not about the fundamental merits of germline gene editing, which in the long run will almost certainly be highly beneficial. Instead, it's about the oversight of science. The concern is that with the technology that are relatively easy to use, like CRISPR, how does the scientific community regulate itself? If there's a silver lining to this cloud, I think it is that the scientific community did pull together to be critical of this work and took the responsibility seriously to use the tools available to themselves to regulate themselves." So Robert here is basically saying that CRISPR is somewhat easy to use and scientists must regulate themselves. And it is good that many scientists across the world saw this issue and all came together and highlighted this issue and critiqued it. Next slide.
Thank you for listening to our topic on gene editing in neurodegenerative diseases. And here's our works cited page with the links to all the sources that we researched about.